and all. Today we are going to discuss about a new topic, which is cytoskeleton. Previously we discussed about cytoplasm, and now we are going to discuss cytoskeleton. What is cytoskeleton? Cytoskeleton is defined as a number or is a filamentous proteinaceous structure of different protein fibers which collectively form the skeletal and structural framework of a cell. So here, cytoskeleton is basically consisted of proteins, and that is uh, also consisting of some particular type of, there are no particular type of organelles. These are only the different types of proteins which are present in the cytoskeleton, which provides shape, support, and movement in unicellular organisms, as well as help in division, cell division, which is what basically provides structural framework to the cell. And that structural framework also helps in movement, as well as helps in cell division, as well as it also assists in several other factors, such as in attachment of one cell to another, but that takes place in particular cells such as the muscle cells, as well as also provides supports to the nerve cells, as well as it also allows the cell to change its shape. And there are lots of other functions, including movement, as well as changing the shape of the cell, as well as it also involves other functionalities such as movement of cytoplasm itself. Cyclosis is also possible by the cytoskeleton. So afterwards, we are having the structure of cytoskeleton, which is present over here over the screen. Here, as you can see, we are having three major types of proteins which constitute cytoskeleton. First, introduce some microtubules. Microtubules are a cylindrical type. This is collectively named as the a set of microtubule, and that microtubule is made up of what? It is made up of tubulin protein. This is made up of tubulin protein, and this is this particular region, this particular column, which is constituting this whole cylindrical structure, is known as microtubule. And inside a single set of microtubule, there are around 13 columns of such microtubules which constitute this whole structure. And microtubules are very important. And what is the diameter of this structure here? So the diameter of this structure here is 25 nanometer. So as it is having a diameter 25 nanometer, it is the thickest protein fiber present inside the structure of cytoskeleton. What is its function? So its function is to produce spindle fibers during cell division. As we know that during cell division, during the process of metaphase, during the metaphase, we are having centrioles. We are having centrioles in which there are microtubules which produce spindle fibers in order to attach with the centrosome. And from which point do they attach with the centrosome? They attach with the point of um, keratosome, and from that point, as they attach from the keratosome, these uh, chromosomes are taken towards the opposite poles. As these are taken towards the opposite poles, that causes to separate the chromosome and turn them into a separate nucleus by forming a nuclear membrane in the upcoming phases of cell division. So I'm generally telling about that process. After which, that also involves what? It also involves um, also involves the division of chromosomes, the movement of chromosomes at opposite poles in plant cells, as plant cells are not possessing centrioles or centrosome, due to which the microtubules which are present inside the plant, inside the cytoskeleton, work as a key factor to produce spindle fibers. As they produce spindle fibers from the cytoskeleton, they basically attach with the chromosomes centromer and bring them towards the opposite poles. So this process takes place in plant cells, while in animal cells, centrioles are also present, in which there are microtubules, but those microtubules are in the arrangement of 9 into 9 plus 0, 9 plus 0. So here 9 represents the side layer of the centrioles, which is composed of 9 triplets of microtubules, whereas the middle part is consisted of 0 microtubules. And as it is consisted of 0 microtubules, it is known as hub. And that will be covered in the topic of centriole, that how centriole and centrosome is formed, and what is the basic structure of it and function as well. Afterwards, we are having some other functions as well of this, such as it being thicker, it allows the cell organelles to be attached at the given position. As we know that throughout the cell, even though the cytoskeleton is fluid, that cytoskeleton which is present in fluid form can cause the cell organelles to float inside it, uh, due to which the cell organelles can just float and cause irregular movement, regardless of the cyclosis which is taking place in the cell. So in this process, the microtubules attach the cell organelles, such as here we are having example of mitochondria. So mitochondria here is attached through this microtubule, as well as through the intermediate filament, which allows this mitochondria to stay in its fixed position whenever the cell is moving through cyclosis. After which we are having intermediate filament, which involves what? Intermediate filament involves its diameter, first of all, is 8 to 12 nanometer, due to which it is considered to be intermediate between the microtubules and microfilaments. That is why these are named as intermediate filaments, and these are made up of what? These are made up of keratin protein, which is present in our hairs and nails as well. And as it is consisted of keratin protein, it is dead filament of cytoskeleton. And what is its function? Its function is to attach the organelles. As I previously mentioned, it attaches the organelles at its fixed position, not allow them to move whenever there is a process of cyclosis. Because inside fluid, inside water or any other fluid, we can see whenever we put any solid inside it, that solid starts to flow. So in this case, it will not start to flow; it will remain attached whenever the process of cyclosis occurs. We are going to discuss how cyclosis will occur as well, and after that, that also provides what? It also provides assistance. It provides assistance in maintaining the shape of cell, and it also provides support to the nerve cells, as nerve cells which possess axons are a bit elongated, due to which it also provides support to the nerve cells which possess axons. After which we are having the third layer, which is microfilaments, which is made up of actin. Majorly, it is made up of two actin chains, which are intertwisted like a in a helical fashion, and these are intertwisted in a helical fashion like the DNA structure. But in actual, the DNA structure, and this is quite different. I mean, in terms of structure and composition, this is completely different. That's only to resemble it with the helical structure of DNA. And alongside that, we are also having myosin, as well as tropomyosin, and we are having myosin, tropomyosin, troponin as well. And alongside that, uh, there is uh, another sort of material inside it as well. That here, these actin and myosin are basically present, where these actin and myosin are also present inside the muscle cells, where these allow attachment of muscles. Here, same case will occur. It will allow attachment, as well as it will also allow the contraction and relaxation of uh, the cytoskeleton, due to which it will cause movement in unicellular organisms such as amoeba, paramecium, are having these microfilaments inside their cytoskeleton, which causes them to move and catch their prey or do anything whatever they want. And that structure uh, is also present in our muscle cells, and that is the reason why our muscle cells are able to contract and relax. And there's a question that why in the muscle cells the contraction and relaxation is more resembled, as well as more prominent as compared to the contraction or contraction inside this cell structure. So the reason is that in muscle cells, the actin and myosin are arranged in a particular arrangement. These are arranged in a proper arrangement, due to which that allows proper contraction and relaxation of muscles whenever we move our muscles voluntarily. After which that also helps in movement of cilia and flagella, movement of pseudopodia and unicellular organisms, as well as that also helps in movement of cytoplasm cyclosis. And afterwards, we are having the diameter of it as well. What's the diameter of it? It is the thinnest filament of cytoskeleton. That is why it is 7 nanometer in diameter. After which we have another point here, that here this intermediate filament is made up of five different types of protein, which includes vitamin. I'm not sure about the spelling, whether it is correct or not. You may just Google it out for the spelling. It's around, I guess it's vitamin, desmin, as well as we are having neurofilament. After which we are having cytokeratin, which is basically this keratin structure, because this keratin structure is mostly considered as this cytokeratin structure is mostly considered keratin. Whenever we are writing generally, so we generally write it as keratin, but here the actual structure present is cytokeratin. And last but not the least, we are having fibrilla. Sorry, it's Giller fibrilla acidic protein. Giller fibrilla acidic protein, and then Giller fibrilla acidic protein, also known as GFAP. And these five types of protein constitute intermediate filament. After which we are having another topic, cyclosis. So how does cyclosis takes place? Cyclosis takes place by means of the motor proteins which are present inside the cytoskeleton. Inside cytoskeleton, we are having motor proteins. Motor proteins which are present inside the cytoskeleton cause movement of cytoplasm, which generate actually force. That force causes to generate cyclosis, cause cyclosis inside the cytoplasm. And that motor protein also causes movement of other organelles such as cilia, flagella, as well as it also causes movement of the complete cytoplasm ultimately. After which, in cyanobacteria, we are having a movement which is known as gliding movement. The gliding movement is done by motor proteins, of course, and that movement is in the form of waves. It is in the form of waves, just as wave glide. It is the same structure just as a waves glide. We are having the same type of movement in cyanobacteria. In amoeba, that movement is known as amoeboid movement, but we call it an amoeboid movement because in amoeba there is less irregular shape. It is irregular shape in general, due to which the gliding movement is not possible. Rather, the mechanism for that is also same, but we call it as amoeboid movement. So this is all about today's video. Hopefully you enjoyed the video, and by now we'll be meeting again in another video.